Hi, my name is Jun Choi, and I'm excited to share my project which involves engineering the bacteria Synechocystis to improve carbon dioxide capture and bioplastic production in high pH conditions. As almost everyone around the world already knows, plastic pollution is getting worse and worse every single year. As you can see from the graph, the amount of plastic entering the ocean has been rising very rapidly. In 2019, there were around 2.3 million metric tons of plastic found in the ocean. To put that into perspective, that is the same as all 10 million cars in California combined. This isn't just about trash piling up, though. Plastic production has a variety of additional effects, including rising greenhouse gas emissions, damage to marine ecosystems, and long-term environmental persistence. The plastics we use today are built to last, and that's the real problem. We need alternatives that are just as useful but far more sustainable. Biodegradable plastics offer a potential solution. One type, known as polyhydroxybutyrate, or PHB, is biodegradable and far more eco-friendly than traditional plastic. It can be made by bacteria like Synechocystis, which is also photosynthetic, meaning it uses CO2 as a carbon source. That means, in theory, this organism can help reduce the rising CO2 levels in the atmosphere and convert it into sustainable plastic. But current solutions have major limitations, since Synechocystis does not naturally make enough PHB for large-scale use. That led me to my main question. Could we get Synechocystis to make more PHB by changing the environment that it grows in? We thought that one way we could achieve this was through adapting the strain to high pH levels. Previous research has shown that at higher pH, more carbon dioxide gets converted into bicarbonate, which Synechocystis can take in more easily. That means more carbon is available for the cells to use. To test this, we started with a method called adaptive laboratory evolution. In this process, we gradually exposed the bacteria to higher pH levels, giving them time to adapt across many generations. After that, we measured how well the adapted strains were growing in a pH gradient. We screened them for PHB using a fluorescence-based dye, and finally, we used RNA sequencing to figure out which genes were changing and why the cells were behaving differently. To evolve the bacteria, we started with a wild-type strain and grew them at pH 7.5. We then sequentially transferred a portion of the culture into fresh media with the pH gradually increased by 0.1 units each time. This gradual change helped the bacteria adapt without dying off. By the end, we had a strain that could survive and grow at pH 11. We also tracked the bacteria's growth throughout the process. Up to pH 9.5, growth stayed relatively normal. After that, it slowed down, but at pH 11, they were still alive and dividing, which was essential for our purpose. So even though growth wasn't as fast as before, they were clearly adapting to the stress. Next, we looked at PHB production using Nile red dye, which binds to PHB fragments and emits fluorescence. So the brighter the cells, the more PHB they produced. The results were clear. Cells grown at pH 11 had the highest fluorescence. We also directly extracted PHB from the cells and confirmed this result. PHB made up over 31% of the cell's dry weight in those high pH cultures, which is 11% greater than PHB made in normal conditions. To understand why that happened, we ran RNA sequencing on the cells. We compared the RNA expression of the evolved strain in high pH conditions to neutral pH, and looked at which genes were upregulated and downregulated, which gave us insight into what was happening behind the scenes. This volcano plot shows genes that were significantly upregulated, shown as yellow dots, or downregulated, which are shown as blue dots. These regulated genes collectively affect biological pathways. Thus, to understand these effects, we did pathway analysis and saw that genes related to photosynthesis and protein synthesis were reduced, while sugar metabolism, especially involving fructose and mannose, were boosted. These sugars help produce acetyl coenzyme A, which is a key building block for PHB, which indicates that the cells were likely shifting their resources towards making more plastic. Overall, we successfully evolved a version of Synechocystis adapted to high pH and producing more PHB. Based on our RNA sequencing, the next step would be to directly overexpress genes in sugar and carbon metabolism to increase precursor availability and enhance genes related to photosynthetic activity to keep carbon fixation high without wasting energy. There's still more to explore, but this is a strong step towards scalable, sustainable bioplastic production.